Okay, so welcome back everyone. This is the solution video for the Delhi University M.Sc Botany Entrance Exam 2020 question paper, and in this video we will be dealing with the solution and explanation of question number 46 to question number 40. So if you want to download this paper, you just have to go to the NTA website, go to their download section and fill the year, exam type and the paper, and you will get this question paper. So starting with the first question, inheritance of Mn blood type is controlled by codominant alleles M and N in a population of 200 individuals. The blood type observed were type M equals to 114 and type Mn 76 and type Nn equals to 10. If the population is in Hardy-Weinberg equilibrium, the frequency of allele M and N will be. So total individuals are 200 and they have given the blood types of M and MM equals to 114, MN 76 and NN equals to 10. So we know that 200 individuals are there for the MN blood group, which can be easily measured since codominance is involved. MN, that is, you can detect the heterozygous also in this. So they are typed accordingly. You can just draw this kind of table. For any such question, you should draw this kind of table. In this, the first column has the blood type, the second genotype, and then the third one is the number of individual. So number of individual divided by the total number of individual gives you the resulting frequency. So 0.57 comes when we divide 114 by 200. 76 divided by 200 equals to 0.38 and 10 divided by 200 equals to 0.05. Now you know the resulting frequency. Just put it into this and the Hardy-Weinberg equilibrium equation. So since mm equals to p square, mn equals to 2pq and nn equals to q square, then p is the frequency of the m allele. So just square root, that is p square equals to 0.57. So p comes out to be 0.76. Now since p plus q equals to 1 in Hardy-Weinberg equilibrium, so just you can minus 1, the 0.76 from 1, that is q equals to 1 minus p, which after which you will get the answer 0.24. So this is the way we do generally the Hardy-Weinberg equilibrium. This is a very simple question on this. I'll just suggest you to just Google the Hardy-Weinberg equilibrium problems and you will get a PDF. It generally has, like on the first page of Google itself, you will get a link where you can download a PDF of a few questions with their answers. And I found that all those questions, all the types of question on Hardy-Weinberg equilibrium are covered in that PDF. If you feel that I should give you a link of that PDF also, then just check the description section of this video. If I remember, I will just put the link of that PDF also. Okay, so next question. Spontaneous mutations in protein coding genes that do not result in change of amino acids are termed as. So answer is synonymous mutation. Let's read a little bit about others also. So transversion in molecular biology refers to a point mutation in DNA in which a single purine is changed for a pyrimidine or vice versa. A transversion can be spontaneous or it can be caused by ionizing radiation or alkylating agents. A nonsense mutation or its synonym, a stop mutation, is a genetic mutation in a DNA sequence that results in a shorter unfinished protein product. That is, it causes a protein to terminate or end its translation earlier than expected. A synonymous mutation, which is our question and the answer. So a synonymous mutation is a DNA sequence change that as a result still does not change the encoded amino acid. Why this happens? This happens due to the genetic code of like redundancy of the genetic code, that is multiple codons code for the same amino acid. Synonym mutation usually occur in the third position of a codon. For example, glycine, the simplest amino acid, is encoded by GGT, GGA, GGC and triple G. So if there is mutation in the third position of the codon, like any of these, it will still result in the same amino acid being incorporated in the protein sequence at that position. Transition, however, refers to a point mutation that show interchange between a purine nucleotide to another purine or pyrimidine nucleotide to another pyrimidine. It can also be caused by oxidative deamination and tautomerization. Next question, a haploid was generated from a plant. The haploid produced perfect bivalent during meiosis. Okay, the plant from which haploid was generated was. So let me tell you a little bit about bivalent. So bivalent is actually one pair of chromosome in a tetrad, and if you see the structure of bivalent, then it is the association of two replicated homologous chromosomes having exchanged DNA strand in at least one site called chiasmata or chiasmata, whatever you pronounce that. Each bivalent actually contains a minimum of one chiasma and rarely more than three. This limited number, I guess, is due to crossover interference, a poorly understood phenomena. We know all that limits the number of resolution of repair events into crossover in the vicinity of another pre-existing crossover outcome, thereby limiting the total number of crossover per homologous pair. So this all details you will easily get in any kind of Snustad-Simmons or Principle of Genetics book. So let's come back to the question. They are saying that actually there is a haploid which forms a perfect bivalent during meiosis. So what was the ancestral plant of that haploid? So the answer is autotetraploid. Let's see a little bit on this. So we actually use the term pseudo haploids for the haploids derived from autopolyploid individuals. So generally, if a haploid is evolved from a autopolyploid individual, they are termed as pseudo haploid, because these pseudo, why they are called pseudo haploid? Because they have a strong tendency to again become a diploid structure. So although it would be hard to establish an existing species as truly autotetraploid, since many so-called autotetraploid species whose polyhaploid have been studied no longer remain strictly autopolyploid, either due to introgation of some genes from the related species or due to a tendency towards diploidization resulting in a great reduction of homology of the chromosome sets by gene mutation. Now many polyhaploids which have their origin from allotetraploid species behave in a way that during meiosis most of the chromosome remains as univalent. So if they are allopolyploid, so like in this case we have the allotetraploid example. So in that case, if they are from the allotetraploid, they have this tendency to be as univalent. On the other hand, there may be polyhaploid derived from the so-called autopolyploid species, or in our case it was autotetraploid species, that shows a very uniform meiosis forming perfect bivalent like a truly diploid form, giving rise to a perfectly normal fertile spore or reproductive propagules, and in this respect sometimes even surpassing the original species from which they are derived. So if they are autopolyploid, and they have this tendency of forming a perfect bivalent. If they are allopolyploid, they have this tendency of forming perfect univalent. Next question, which one of the following statement is not true about isochromosomes? The answer is formation of isochromosomes generate deficiency loop. Yes, it is wrong. Let's read a little bit about it. An isochromosome is an unbalanced structural abnormality in which the arms of the chromosomes are mirror image of each other. The chromosome consists of two copies of either the long arm or the short arm, because isochromosome formation is equivalent to a simultaneous duplication and deletion of genetic material. Consequently, there is partial trisomy of the genes present in the isochromosome and partial monosomy of the genes in the lost arm. So you can just check the other options also with this explanation. Isochromosomes can be created during mitosis and meiosis through a misdivision of the centromere or U-shaped strand exchange. Isochromosome do not form deficiency loop. So that option, the deficiency loop option, that is the second option, is actually wrong, because they do not form deficiency loop. Rather, they are generally formed by deletion, duplication or inversion. A deficiency loop in particular denotes deletion. Fine. So deficiency loops are not formed by isochromosome. They are generally formed by deletion, duplication or inversion, and in most cases they are more like specific when there is a deletion mutation. Next question, which is the mode of inheritance in the pedigree given below? Okay, so yahan like in here we have this thing, a lot of information is giving already. The male is infected, and then in the next generation some of the kids are infected, and from one infected female we have the another generation having one infected female again. So actually this type of pedigree is an autosomal dominant pedigree. Let's read the characteristics of autosomal dominant so that you can come out for this option easily. So characteristics of autosomal dominant inheritance: male and females are affected in roughly equal proportions. Pupil in more than one generations are affected. Men and women are both able to pass on the condition to their sons and daughters. Only one copy of disease allele is necessary for an individual to be susceptible to expressing the phenotype. With each pregnancy, there is 1 in 2, that is 50% chance the offspring will inherit the disease allele. Unless a new mutation has occurred, all affected individuals will have at least one parent who carries the disease allele, and male to male transmission can be observed. Example of disease with autosomal dominant inheritance includes myotonic muscular dystrophy and Huntington's disease. So if you just check all these characters in this pedigree, you will easily come to know that it is an autosomal dominant types. Next question, fill in the blanks with the option given in the correct order. The Shine-Delgado sequence. So this is the first lecture of molecular biology. If you have gone through my lectures, so this was the first lecture which I took, where we discussed about Shine-Delgado sequence, Kozak sequence and all. So okay, the Shine-Delgado sequence pair with dash present in dash. So I'll just read out. The Shine-Delgado sequence in a ribosomal binding site in bacterial and archaeal messenger RNA, proposed by Australian scientists John Shine and Lynn Delgado. These sites are also present in some chloroplast and mitochondrial transcripts. It is a 6-base consensus sequence AGG AGG. In E. coli, it is actually AGG AGGU. Generally located around 8 base pair upstream of the start codon, AUG. The RNA sequence help recruit this Shine-Delgado sequence actually helps to recruit the ribosome to the messenger RNA, and so that a protein synthesis can be initiated by aligning the ribosome with the start codon. So you can see it actually paired with the 3' end of the 16S rRNA, and this whole 16S rRNA is actually present in the 30S ribosomal subunit. So the option you can easily deduce which option is correct after this explanation. Okay, so next question, transcriptionally active regions in eukaryotes are known to be dash and generally carry a dash mark on the histone protein. So we know that there are two types of region in the chromatin. The transcriptionally active regions are called as euchromatin, while the transcriptionally non-active or silent regions are called as heterochromatin. So let's read a little bit on them. So covalent histone post-translational modification, that is HPTMs, such as acetylation, methylation, phosphorylation, ubiquitination, sumoylation, ADP ribosylation, deamination, and the non-covalent proline isomerization can affect the condensation of chromatin as to organize the genome into transcriptionally active, that is euchromatin, or inactive regions, that is heterochromatin, respectively, and recruit effector proteins. So euchromatin, as the question is asked, is asking about this the types of this S3K types of protein. So euchromatin is characterized by high level of acetylation and high level of S3K4Me123, S3K36Me3, S3K79Me123. On the other hand, heterochromatin is characterized by low level of acetylation. So actually there is hyperacetylation in the euchromatin, while in the heterochromatin there is low level of acetylation, that is hypoacetylation, and there are high levels of S3K9Me23, S3K27Me23, and H4K20Me3. So you just have to just make a table, and on the left hand side just write euchromatin, on the right hand side write heterochromatin. Euchromatin you write hyperacetylated, heterochromatin you write hypoacetylated, and just write the name of these proteins below that. When you will draw the table, it will be easy for you to remember. Next question, the dinucleotide sequence that marks the 5' and 3' end of exon intron junctions are. So this is actually when you will read the splicing, alternative splicing, the whole spliceosome formation complex, you will come to know about this axon-intron junction. I'll just show you the thing. So in the red circle, these are the axon-intron junctions. So there is actually no extensive homology that exists between the end of an intron, but the intron-axon junctions though do have a well-conserved short sequence, as you can see. And the question, whatever the question is asked, is actually marked in that red arrow. The GT and the AG respectively are present at the 5' and 3' end of the axon-intron junctions. Next question. A common outcome of infection by plant viruses like TMV is likely to be. Okay, so this is, they are asking about the symptoms, the causal symptoms. The organism is TMV, that is tobacco mosaic virus. So what is the common outcome of infection? So all of these, out of these options, the reduced production of Rubisco is infected cell is correct, because a lot of study recently has been done on this Rubisco interaction with the proteins of tobacco mosaic virus. Let's read a little bit on it. So the multifunctional movement protein of tobacco mosaic virus, we can, in short, we call it as tobamo virus, is involved in viral cell-to-cell movement. We know that all the these viruses have this movement protein encoded by their genome. This protein actually interacts with the various proteins of the plasmodesmata so that they can easily be transferred from one cell to another cell. So again, I'll read just the multifunctional movement protein of tobacco mosaic tobamo virus is involved in viral cell-to-cell movement, symptom development, and R gene recognition. So tobacco mosaic virus MP, that is movement protein, has three functions. It actually does the viral cell-to-cell movement, it causes the symptom development, and it is also involved in resistant gene recognition, the resistant gene of the host. So Rubisco catalyzes the first step of CO2 assimilation in photosynthesis and photorespiration. We all know that. The Rubisco holoenzyme is a heteropolymer consisting of 8 large subunit RBCLs and 8 small subunits RBCS. So small subunit is denoted by S, the larger subunit is denoted by L. So RBCL was reported to interact with the coat protein of potato virus Y. This is a very important research and very keystone research from a very reputed lab in Europe. Both RBCS and RBCL were reported to interact with the P3 proteins encoded by several potyviruses, including shallot yellow stripe virus, onion yellow dwarf virus, soybean mosaic virus, and turnip mosaic virus. So RBCS plays an essential role in virus movement, host susceptibility, and TM22 mediated extreme resistance in the tobacco mosaic virus host plant interaction. So tobacco mosaic virus MP interacts with the Rubisco small subunit of Nicotiana benthamiana both in vitro and in vivo. So after reading this explanation, I guess you will be easily understood why the TMV infection causes the reduced production of Rubisco in infected cell. Actually, they have there are a lot of reports where they have shown the interaction of this movement protein and the Rubisco smaller subunit. And recently it was established in Nicotiana benthamiana both in vitro and in vivo. But yes, I'll accept this, that this was a tough question. Like if you are not going through research papers and all, it is generally not written in the textbook that where this virus is actually infecting. Textbook of your undergraduate level. In the master's level textbook, yes, like if you read the book of plant pathology by Agarrios or this plant virology by Roger Hull, you will easily answer this question. But yes, it is a tough question for an undergraduate level aspirant. Okay, so next question. Translation of multiple polypeptide from a single mRNA does not involve. Actually they are talking about leaky scanning, and leaky scanning is a phenomena in which a weak initiation codon triplet on mRNA is sometimes skipped by the ribosome in translation initiation. The 40S ribosomal subunit continues scanning to further initiation codon. The weak initiation codon can be an ACG or an ATG in a weak Cossacks consensus context. This way, mRNA can encode for several different proteins if the AUG are not in frame, or for proteins with different N-terminus if the AUG are in the same frame. So let's just read the options. So the question was, translation of multiple polypeptide from a single mRNA does not involve. So number one, leaky scanning by some eukaryotic ribosome. Yes, it is correct. It is actually there. Then there is protein translation from only the first AUG codon. No, wrong, because if it is from the only from the first AUG codon, always the same frame will be read, and hence always only a single kind of polypeptide will be formation. Multiple polypeptide formation will not be taking place. So rest other you can easily understand by just reading the paragraph at the down. So the second option, that is the protein translation from only the first AUG codon, is wrong. Yes, it is very important to note that leaky scanning is actually observed in various viruses, especially the plant viruses. So several viruses use a leaky scanning mechanism to produce vital proteins, which implies that leaky scanning is not a consequence of inadequacy, but instead allow viruses to overcome the high selective pressure of the competing with their host. So actually leaky scanning, as a, like if the name you just read leaky scanning, you feel like okay, something is something mistake is happened in the scanning of the ribosome when the ribosome scan the mRNA. No, actually this is not a mistake. In fact, some of the viruses actually use this leaky scanning mechanism to produce multiple protein at a time. Why they need that? Because they have to defend them from the host resistance. Like these viruses either be acting to the plant or an animal. To defend themselves against the host defense mechanism, these viruses need to replicate faster and make multiple copies of their protein, in which this leaky scanning phenomena helps them. So it is actually a natural phenomena that occurs in viruses, and it's frequently observed in viruses. Otherwise also in other systems also it is observed. Next question, the absence of 5' cap structure in many plant viral RNA is not associated with. So a cap independent translation element, CITE, that this question is actually asking question on CITE. So a cap independent translation element, CITE, or 3' site, is an RNA sequence found in the 3' UTR of many RNA plant viruses. Many viral mRNA lacks the 5' cap. Animal virus mRNA often contain an internal ribosome entry site which function in translation initiation. Many plant viral mRNA contain a cap independent translation element. These elements mediate initiation of translation of the proteins encoded in the mRNA by either recruiting the translation initiation factor or the 60S ribosomal subunit to the viral RNA. There are many different structural classes of cap independent translation element with no apparent structural or sequence similarity. So the last line says that there are many structural classes of cap independent translation element with no apparent structural or sequence similarity. So yes, the first option says that it is highly conserved 100 nucleotide long cap independent translation element. No, it is not highly conserved. There will be various structural classes, and there is no sequence similarity or apparent structural or sequence similarity between the two cap independent translation element. So the option first one, the high conserved 100 nucleotide long cap independent translation element, is the wrong one. Okay, carrying capacity is a characteristic feature of. Okay, so carrying capacity, you know that how much environment can carry the pressure on it due to the biotic and abiotic factors, unless this environment get disbalanced. So this is what a carrying capacity is. Let's read a little bit about it properly. So in ideal environment when there is no limiting factors, populations show a smooth and steep exponential growth curve. That is, you can see the orange one. However, under normal condition, factors such as limitations in food, competition for other resources, or diseases are present. We know that normal condition is like this only, that when there is a limitation or competition for food, shelter, mate, resources, and there is disease also. So gradually in a population, the competition increases and resources become increasingly scarce. Thus population reach the carrying capacity of their environment. So carrying capacity is the maximum amount of the tolerance threshold the environment have to properly carry a population or a biotic community. This causes their growth rate to slow rate. Like when the population reaches the carrying capacity, their growth rate starts slowing down, and almost become nearly to zero. This produces an S-shaped curve of population growth known as logistic curve or sigmoidal growth curve, which is shown in blue. Okay, next question. Plants such as prosopis, acacia and capparis are more likely to occur in. So there are four options. The question was, I just repeated, the plants such as prosopis, acacia and capparis are more likely to occur in so tropical thorn forest, deciduous forest, evergreen forest and grassland forest. So the answer is actually tropical thorn forest. So tropical thorn forest or tropical shrub forest are forests having intermediate moisture conditions between a desert or savanna on one hand and a seasonal or rainforest on the other hand. Acacia and prosopis are perennial non-succulent plant of such region, while capparis is a xerophytic shrub which is found there. Next question. Ecosystem resilience is known to be maximum in. Ecosystem resilience, if we say, is the way an ecosystem responds to a perturbation or disturbance by resisting damage and recovering quickly. So that how conveniently an ecosystem responds to a disturbance and how quickly it recovers from that. So perturbation can be anything like natural such as fire, flooding, windstorm, insect population explosion, or anthropogenic, that is man-made, such as deforestation, fracking of ground for oil extraction, pesticide sprayed in soil, and the introduction of exotic plant or animal species. So these disturbances may change the ecosystem if they cross the threshold limit of the ecosystem resilience index. So there is an resilience index also in the ecosystem. So if that resilience index is crossed, so ecosystem is not able to recover. So like in this four option, the maximum resilience is found in grassland, then forest, then tundra and desert have equal amount of resilience. So I've just kept them at the end and then equalize between them. Okay, so next question. Champion and Shade classified the forest type of Delhi as. If you just Google the Champion and Shade 1969 and just see the Google images, like if you are not able to see this image properly or there is a resolution problem with you with your cell phone or laptop in which whatever you are seeing, you just Google Champion and Shade 1968. You will get this image in the Google images. This is the map of India where they have classified the various places according to the various forest type. And if you see it properly, so the Delhi has the tropical thorn forest type of vegetation. You have to just see this map once with the various guidelines which are giving in the map. You will come to know which area is classified under which forest type. Next question. This was the last question, I think. So in the next video we will have the explanation of question number 61 to question number 75. If you find any mistake in the solution, please do comment. Please like the video, comment and subscribe to appreciate my work, and you can also share this video among your fellow aspirants. And please stay tuned for more upcoming solution videos of various MSc entrance exams related to biological sciences.